I will move a little bit from the liver down to the intestine, which I think is a very intriguing side to discuss some data regarding nutrigenomics, because for me, the intestinal mucosa is one of the most important first pass for getting in contact with nutrients. So in the first slide, I'll try to introduce the intestinal mucosa development. And as you know, on the left part, we are in the ileum, and on the right part, we are in the colon. Here is depicted only the cells that give rise to intestinal mucosal epithelium. And as you know, on the bottom of the crypt, there are 3 to 4 intestinal specific stem cells that give rise to a particular type of cells that are called proliferative progenitors in the transit amplified compartment of the crypt. And those cells, the only thing that they have to do is mostly proliferate and eventually start migrating up to the villus axis. At a certain moment, they reach a kind of non-anatomical but functional barrier where those proliferative progenitors, they block completely their proliferation and they differentiate into the mature enterocytes, which are the one we are used to talk when we discuss, for example, fatty acid metabolism or cholesterol absorption or glucose absorption in the gut. So what is happening to these mature enterocytes in the villi or in the surface epithelium of the colon? Actually, they live for 48 or 72 hours. They make most of the action that we look forward to for the interaction with nutrients. And then at the end, they die for apoptosis. So the main important thing and why I got interested in this type of field is because the first thing we need to know is who are the main player in modifying the metabolism of those cells. And of course, the main players are transcription factors. And eventually, there are several nutrients that are able to modify, to induce, to give speed, or to delay such of a metabolic pathway. The importance of the proliferation and differentiation is also because the tumor formation depends mostly on mutation on those cells. And the recent data from the laboratories of Hans Clevers and Mario Capecchi have clearly shown that the mutation that give rise to cancer are probably coming mostly in the stem cell compartment, where a single mutation that normally changes completely the identity card of those cells will allow these cells to continue the proliferation and therefore to have a special type of metabolism. So how is starting the colon cancer formation? And this is a classical slide that was depicted mostly by Professor Wolgenstein in the US. The main mutation for colon cancer, we are talking also of sporadic colon cancer. Actually, 85% of colon cancer in the population have an APC mutation, so a mutation into the system that regulates the shuttling of the beta-catenin in the nucleus that drives the proliferation. And then after that mutation, several other mutations are accumulating until from a preliminary dysplastic lesion, you reach a clear carcinoma with invasion of tissue and metastasis. And mostly what is happening here is that those cells are able to escape completely the normal apoptotic pathway that is the normal things that happens into the enterocytes. Now, those cells have a complete change in the metabolism. And the main change that they have is mostly that when there is a transformation into tumor cells, they mostly rely on glucose for their metabolism because they are used to work into hypoxic condition, and we heard this morning how this is working. But the intriguing thing is trying to understand who are the transcription factors that get the modification and how is possible to bring eventually them back to a normal, differentiated, mature type of metabolism. So our goal is not to try to avoid a mutation. Our goal is that, let's say that the cell is mutated and changes metabolism. Now, we don't think that we have to bring back the normal gene into that cells. What we think is that via nutrients or eventually nutraceuticals, we can change the metabolism of that mutated cells and bring it back to a mature type of metabolism that will block the growth of the cells. And to do that, we work mostly on the nuclear receptor superfamily. This is one of the largest family of transcription factors in our genome. These are 49 guys. The first one was cloned in 1985 by Ron Evans. It was the glucocorticoid receptor. And from that point on, for sequence analogy, the other 48 members have been cloned. And when we talk about nutrigenomics, when we talk about the way our nutrients, or eventually nutrients-derived molecules, can switch on and switch off gene, I think it's very important to discuss about this family of transcription factors, because as you can see, most of the vitamins or the hormones or the fatty acids or cholesterol derivative oxysterols or any xenobiotics are able to shuttle to the nucleus to bind the ligand binding domain of these guys and then to change the conformation and induce the transcription. And for highly frequent type of disease, I think it's much more important to focus on the way we interact with the environment than to try to figure out a single gene mutation that could characterize why we are susceptible to cancer or to obesity. Of course, the first thing that one can do is try to identify where are these transcription factors, where are they expressed, what is their cell type specificity, and especially if there is a pattern of expression, this could be eventually translated to a pattern of function. And we did this study recently for the intestinal mucosa. We could really figure out that there are some of these proteins that are in the differentiated cells, some that are in the crypt, and some that are in the stem cells. And if they are there, Mother Nature made them being there because they probably are driving some of the most important function in terms of transcription. Now, how do they work? Actually, there is a ligand that arrives, and when the ligand arrives into the nucleus, these guys are normally on the promoter of their target gene. They have a coreprexor complex that is bound directly to the 2 protein, and the chromatin structure is into a repressive status. When a ligand is arriving, it's changing the conformation of those proteins, especially on the ligand binding domain, and then the corepressor complex goes away, and now co-activators are arriving, the transcriptional machinery is recruited, and the genes are on. Of course, this is a cartoon. This is a dynamic thing that is probably happening several times during the hours in our body. Now, talking about co-activator, the talk of today will be focusing on this guy, PGC1, which was already mentioned by a previous talk. PGC1 alpha is actually a transcriptional co-activator. It's not a transcription factor because it does not bind directly DNA. It needs some protein that are on the promoter to induce the transcription. And those proteins are mostly nuclear receptor, but also other non-nuclear receptor transcription factors such as nuclear respiratory factor 1 and 2. PGC1 alpha was discovered and cloned by Bruce Spiegelman in Harvard a few years ago and is highly expressed in all the high energy demand tissues. So what is known regarding PGC1 alpha in our body? Let's say that the most important discovery that Bruce Spiegelman did was that when we are exposed to cold, we have the classical adaptive thermogenesis in the brown adipose tissue, at least in a mouse, in humans in the skeletal muscle, or eventually in the brown adipose tissue, according with the latest paper in the literature. And there, PGC1 alpha was discovered because via a big transcriptional network is able to drive several things, which are the same things that go on during exercise in the muscle or during fasting, for example, in the liver. And mainly what it does is mitochondrial biogenesis. And together with biogenesis, it increases mitochondrial function, it increases the respiratory capacity, and it makes fatty acid oxidation. Now, this is the knowledge regarding PGC1 alpha till a few years ago, and together with this, we know that there are several other important things such as the uncoupling and the gluconeogenesis that are cell type specific. Now, our goal was to try to understand if PGC1 alpha is expressed in the gut. And for the first time, we did a classical array for the expression, and we found out that from the duodenum till the colon, there is a quite high expression of PGC1 alpha. And you can see there is a cycle time of 21 for the real-time qPCR aficionados in the audience. And when we go to look where PGC1 alpha is expressed, and here is an immunohistochemistry of the colonic mucosa, you can clearly see that the important thing is that PGC1 alpha has a specific expression pattern localization, which is mostly in the differentiated cells of the surface epithelium in the colon or in the villus axis in the intestine. There is also some scattered expression lie down in some stem cells, but this is not going to be the topic of today. So PGC1 alpha is expressed selectively in the differentiated cells, which are those cells that they have to breed, and they have to work, and they have to die for apoptosis. So what we did, let's look at what is happening to PGC1 alpha in cancer. And we took APC mean mice, which are mice with a germline mutation, which make them susceptible to make a somatic mutation and then colon cancer. And we have the same patients with our called familial adenomatous polyposis patients that have exactly the same mutation. We were able to get through a collaboration with Renato Mariani Costantini the samples from the human subjects. And when you go to look at the transcript, there is a 50% decrease in PGC1 alpha transcript in tumors of mice, and the same in humans. And this happens also with their target. Here is depicted MCAT, which is an important protein in the fatty acid oxidation pathway, but the same is happening for the mitochondrial biogenic protein. So our next step was, if PGC1 alpha is decreased in cancer and is expressed only in the differentiated cells, we said let's force its expression in colon cancer cells and see what is happening. So we use 3 different types of colon cancer cells. I will drive you through this slide. The intriguing thing is that colon cancer cells are mostly driving their metabolism via glycolysis. And they make ATP via mostly anaerobic glycolysis. You can culture them in hypoxia, they will grow up even faster. So when you overexpress PGC1 alpha, the first thing that happens, you have a double amount of mitochondrial DNA, which means double amount of mitochondria, and we have the labeling with the mitotracker that clearly tell us that there are much more mitochondria. So we now have colon cancer cells with a double amount of mitochondria. And the thing is, are those mitochondria functional? What we did, we studied the oxygen consumption in those mitochondria, both the coupled and the uncoupled oxygen consumption. As you can see, there is a strong increase in the consumption of oxygen in those cells. So the colon cancer cells now have double amount of mitochondria, they breathe more, so the respiratory capacity is increased. But the intriguing thing is that they get a very large increase in apoptosis. And this is seen via Nexin-5, but also via several other staining that we did. In order to understand why apoptosis was coming in these colon cancer cells, what we did, we studied the production of ROS. And the reason why we did that is because when you activate the number of mitochondria, their function, surely you're increasing respiratory capacity and you might produce more ROS. And indeed, if you look at this panel, there is an incredible amount of ROS that are formed, or let's say that are accumulated in intestinal cells, but this is not happening in the muscle cells. And this goes together with the nucleosome enrichment. So we increase the number of mitochondria in cancer cell, we increase respiration, we have ROS production or accumulation, and we do have apoptosis. So we will leave this discussion in a few slides. We then took this slide and we put it into a xenograft mouse in order to study if this was true also when you study the cell growth in vivo. And we did a classical tumor volume. As you can see, there is a delay in the growth of tumor when you inject via adenovirus PGC1 alpha versus the control LAC-Z under the skin of the xenografted mice. And if you take out the tumors and you study them, well, of course, you see more PGC1 alpha, but also if you do the activity of the cytochrome oxidase, which represent actually the respiratory capacity in vivo, you can see that there is an increased respiratory capacity, there is increased ROS production, and there is increased tunnel staining, which means that we are actually having apoptosis in those colon cancer cells, and we are able to decrease the tumor growth. Now, at this point, we said there is more mitochondria, there's more ROS, but is oxygen needed for that? And one way to study if oxygen is needed is to modify the hypoxic condition, like we heard this morning. But actually another way is to not changing the oxygen, but now deleting completely the mitochondrial DNA. These are cells that are called Rho0 cells that have been generated by a collaborator, Gaetano Villani, who studied it in the laboratory of Professor Attardi, which were the first to generate the Rho zero cells. These are cells without mitochondrial DNA. So we took colon cancer cell line, we delete completely the mitochondrial DNA. As you can see, there is no labeling, there's no level for cytochrome B. And when you study oxygen consumption, there is no oxygen consumption. So now you have oxygen, but there is no consumption because the mitochondrial DNA is out. And what is happening? When you overexpress PGC1 alpha, the transcriptional machinery goes on. So the target gene in the nucleus are all activated, but now there's no ROS production in the Rho zero. And there is no apoptosis. So this proves two things. Number one is that you need a mitochondrial to breed and to produce ROS. And number two, that the apoptosis that PGC1 alpha is inducing in colon cancer cell is driven via a complete mitochondrial pathway. Because if mitochondria are not there, you don't make ROS and you don't make apoptosis. The second thing is that we went back to the literature, and there is a nice study by Scarpulla and Spiegelman himself, which shows that PGC1 alpha is actually able to protect from ROS formation in high energy demand tissue, such as, for example, muscle. And what we did, we went back to those cells that I showed you at the beginning, which are two cell lines that are colon cancer cell, but we also used the C2, C12, which are the actual cell line that those guys used in the study to prove that PGC1 alpha was able to protect from ROS production. And one thing that we saw is that the transcriptional induction of the scavenger system, actually the superoxidase dismutase, the manganese one, and the catalase is induced in the muscle, but is not induced in the colon cancer cell. So the reason why in colon cancer and probably in other cancer cells, PGC1 alpha increase mitochondrial respiration and make ROS is probably due to the fact that in those cells it is not able to upregulate the transcription of the scavenger system that normally gets rid of the ROS formation. And that would make sense because the scavenger system, in a way, in cancer have been involved on, from several years, not only for that, but also because if you go in vivo in intestinal mucosa, the upper cells, the enterocytes, have a very low scavenger system. Indeed, after 48 hours, they die because they start breathing, they make ROS, and they have very low SOD2 and CAT expression, which on the contrary is very high in the stem cell. So in the last 5 minutes, what I will show you is that if you put a mimetic of SOD2 and catalase on those cells, which is called UC134, you can clearly block the ROS production via PGC1 alpha, and you also block the pro-apoptotic effect. And if you now go in vivo and you inject the cells and the tumor into the xenograft model, and you do again with the PGC1 alpha, with and without the SOD2 and catalase specific mimetic, you can see that the effect of PGC1 alpha on the delay of the tumor growth is completely blocked. So these data prove that the pro-apoptotic and the respiratory capacity driven effect via PGC1 alpha in the gut is probably due to the absence of the transcriptional capacity of PGC1 alpha to induce the scavenger system. What we did then, we tried to go in vivo, because all the data that I show you now are on colon cancer cells, and I know that most of you were preparing questions regarding how this is important in terms of physiology. So what we did, we overexpressed PGC1 alpha in the normal intestinal mucosa, and to do so, we generated a mouse which overexpressed PGC1 alpha under the control of the Willing promoter. And what is happening is now we have the human PGC1 alpha which is overexpressed throughout the entire gastrointestinal tract. And when you look at the protein, as you remember, PGC1 alpha was only on the top and is now in the entire villus, cryptovillus axis. So this is a transgenic mouse that overexpressed PGC1 alpha, and we were expecting a phenotype. And what happened to this mouse is that if you do electron microscopy of the apical enterocytes, you can clearly see that the number of mitochondria is extremely high, and actually there is a big change into the type of mitochondria, but this is something for the cell biology aficionados. The important thing is that if you go to function, the number of mitochondria is increased, but also the respiratory capacity of the upper intestinal enterocytes is increased. And this goes together with ROS accumulation and apoptosis. So it is exactly the same that happened in the colon cancer cells. This means that if we are able to change the amount of the respiratory capacity and the mitochondrial biogenesis in the gut, but we are playing with the lifespan, or if you prefer, the aging, and eventually this could be important for cancer formation. In fact, if you look at the morphology of these guys, you can see that the transgenic mouse have a very low villus as compared with the normal mouse. And if you take the knockout, they have a very high in terms of length of the villus, and the same for the colonic crypt. And this is quantified here, and it's also quantified in terms of the tunnel positive cells. So we are playing with the aging of the enterocytes, and surely any nutrients that would work on PGC1 alpha would do so. Is this important for cancer? So we did two models of cancer. One is a chemical model, which is a chronic colitis via giving a carcinogen, AOM and plus DSS on several. And also we did a genetic model, which is we crossed these mice with the APC mean mice. And I conclude by showing you the data on tumor formation. This is a work of 4 years in the laboratory, and as you can see, the number of tumors when you overexpress PGC1 alpha is significantly lower, especially in the ileum, and who knows about mice knows that APC mean mice have a tumor, especially in the ileum, the polyps, but also the volume is much lower. Intriguingly, when you study that in the knockouts, you have the vice versa phenotype, that the tumor volume is increased and the number of tumor is also increased as compared with the wild type. So what I told you today is that if you increase the amount of breeding capacity, if you increase the amount of mitochondria, if you let a tumor breed, you will probably block its growth. And indeed, when we were asked how is happening that PGC1 alpha is blocking tumor growth, because you have a mutation, so what could happen? And actually, if you take out the tumors from the transgenic mice, you can clearly see that inside the tumors, PGC1 alpha is overexpressed, and that's because vilin is an integral part of the stem cell, therefore drives the expression also in the tumors. And when you go to study, you see that the apoptosis is coming on in the tumor. And this is exactly what we had in the xenograft model. So I do think that if we are able, via nutrients modification, to make the mitochondrial being more in number and being more functional, which are two different things, mitochondrial biogenesis and mitochondrial function, we will definitely be able to block cancer independently on the mutation that was the first on starting cancer. So the take-home message is that PGC1 alpha is a transcriptional co-activator and is able to upregulate mitochondrial biogenesis, OXPHOS, and fatty acid beta oxidation, and the final aim is providing efficient pathway for aerobic energy production. Now, in the intestine, there are glycolytic cells that are the proliferating cells in the crypt. They migrate along the villus axis and they differentiate into mature enterocytes, and their destiny is to die for apoptosis within 48 and 72 hours. Now, PGC1 alpha is expressed in the intestinal surface. It drives mitochondrial biogenesis and respiration in presence of reduced antioxidant enzyme activity. And this determines the accumulation of ROS and fosters the fate of enterocytes toward apoptosis. Thus, we show you today that we had gain and loss of functional approaches both in cells and mouse model, and we presented this scenario where PGC1 alpha is able to regulate the cell fate and protects against tumorigenesis. If you want me to make some speculation of nutrients, well, you probably know that the function and the activity of PGC1 alpha is regulated via sirtuins, and therefore it's only a speculation which one could be the nutrients that should be used, and some of those are probably already used as anti-cancer strategy. Let me conclude by thanking the person who did this work in the last 5 years. Ilenia, she was quite talented. She's now for a postdoc for 1 month in Switzerland, in the Walter Valley laboratory. And these are all the collaborators that gave us help and support throughout the years, and the Italian Association of Cancer Research and the European Research Council for the grant support. Thank you.